Hello, everyone. Thank you very much for being here today, this morning, if you're watching this live. My name is Jeremy Weiss. I am a professor of biology at Linfield University, and I am a Oregon Wine Board funded researcher. So I'm going to be talking with you this morning about some of the research that I've been doing on terroir and microbiomes. I want to start with some quick acknowledgements. This is I lead this project, but this project could not happen without the help of many Linfield University students. So I have their names up on the screen and some pictures for you to see. And a huge thank you to all the vineyard owners and managers that let me come out onto their property and harvest a few grapes and take some samples and chat with them. And so I really appreciate all of that time and effort that those individuals have provided me. All right. Oh, and I forgot, of course, that, as I said, this is funded by the Oregon Wine Board as well as by Linfield University. So I want to jump right in. I gave a talk at the symposium a couple years ago on this, so I'm not going to do a lot of background, but just a quick reminder that there have been a handful of studies looking at microbiomes, all of the microbes, bacteria, fungi, yeast, things like that, that we see in vineyards. And the one sort of big study that was sort of most conclusive showed that geography influences the vineyard microbiome. So I realize this is a confusing figure, but really what you just have to see is that from samples from the Central Coast in California versus Napa versus Sonoma, that the microbes that are present in the vineyard are very different depending on what location you're in. So that sort of set the foundation for all the work that we've been doing. But it's not just geography, right? Terroir is not just geography, it's all sorts of things. So the big sort of question that we've been asking is, do viticulture practices, so the kinds of things that you or your vineyard managers do, influence the vineyard microbiome? And is that more or less than the geography? So a real quick note, for the purposes of this presentation, we're defining biodynamic vineyards as those with Demeter certification, and I'm broadly classifying everything else as conventional, though a lot of the vineyards that we sampled in our conventional class are really live certified. So we go out to vineyards and we collect some clusters. The way we've been doing this is one cluster of grapes per row, and we collect every other row until we get to 5 clusters per block. And that constitutes our sample from a vineyard. So we go out, we do this about 2 weeks before harvest. We take the grapes back to Linfield and we take 4 berries off the cluster, kind of evenly spaced out. We mash all those berries together and we do DNA extractions on them, send that DNA to a sequencing facility at Oregon State, get data back, and we get huge amounts of data back. So for this project, I have about 20 gigabytes of data. It's about 100 samples and about 1,500 species. And then we take that data, and I will show you in a second what we found. But we are using AVAs as our measure of geography. So these are AVAs in the Willamette Valley, and specifically, we have sampled from vineyards in 5 AVAs. And these samples are all from 2017 and 2018 vintage grapes. So we sampled from Ribbon Ridge, from Yamhill Carlton, from McMinnville AVA, from Dundee Hills, and from Aola Amity Hills. And just to show you real quick, right, we have from each of those AVAs, we sampled a biodynamic, a Demeter certified vineyard and a conventional vineyard. And we tried to pick things that were geographically close to one another so that terroir would otherwise be similar. So these are the vineyards. We gave them all abbreviations. If you're familiar with these areas, then you probably know what the vineyards are, and that's fine. If you're not, and you're really curious, then I can share that information with you afterwards. Okay, so when we do this, we get data on the community of microorganisms, and I separate this into bacterial data and fungal data that's primarily yeast. What you're seeing now is data that I have shared in the past. So I put this together, but I just want to remind everybody what we found thus, what we had previously found, and also a quick refresher or understanding of these plots. We take all of that data and we in terms of the abundance and diversity of microbes in a sample, so off a cluster, and we do a statistical analysis that allows us to put that in one point in two-dimensional space. So each point here represents a sample, and then I connect all the samples together that are the same. So the outer circle here are bacterial data from conventional vineyards. The inner one here is bacterial data from biodynamic vineyards. You can see there's a lot of overlap, but they are significantly different. So bacteria communities are different in biodynamic vineyards versus conventional vineyards. If we take the same data and break it up by AVA, you can see it's just a jumbled mess. So we're not seeing differences in bacterial data between AVAs. If I take the same data and break it up by vintage, now the 2018 samples are significantly different from the 2017 samples. You can see that these two polygons don't overlap at all. So our biggest factor in determining bacterial communities on grapes is not actually farming practices or geography, it's actually vintage. Now, the fungal data, right? This is totally new stuff that I've not presented yet. And what we found is that there are no significant differences based on farming practice. So if you look at the biodynamic vineyards versus the conventional vineyards, the communities are, there's variation in communities, but there's a lot more variation sort of within a sample than between these samples. So what yeasts and other fungi are present on grapes does not change if you're biodynamic or not. If we take that same data and break it up by AVA, now we're getting significant differences. So the McMinnville AVA is significantly different from the Amhill Carlton. Ribbon Ridge is mostly separate. Interestingly, Dundee Hills and Aola Amity overlap a lot. And I say that's interesting, right? Because those are geographically not next to each other, but we're seeing some fairly similar fungal data there. Okay, so what, right? So this is some data, and again, I'm happy to chat about this with people at any point, but what does this mean? The biggest takeaway that I want you to get from this is that the microbiomes of wine grapes are highly variable, both in space and in time. And while that variation is driven largely by geography and vintage, farming practice does play a role in it, right? It's not overwhelming, and it depends on whether we're talking about bacteria or fungi, but it does matter what you do. What happens in your vineyard influences what bacteria and what yeast you can find. So to sort of reiterate that point, right, microbial terroir, the impact of these microbes on the wine that you get is very likely influenced by things that the vineyard owner can control, right? It's influenced by geography, it's influenced by vintage, which presumably is weather, climate issues, but it's absolutely influenced by things that you can control. So, right, if these are things that you want to change, then you can change your farming practices and get different microbes there and different microbes into your winery. So this is, as I said, this was data from 2017 and 2018. In 2019, Dr. Krista McGuire from the University of Oregon and I collaborated, and we sampled 12 vineyards across the state. So not just in Willamette Valley, but in Southern Oregon AVAs, and as well as in the Gorge. And we're working on correlating those differences. Unfortunately, our work has been delayed by the pandemic. So we're still trying to process through our 2019 data, and I hope to share that with you soon. But at this point, I don't really know what soon means. So, all right, that was a very quick run through of my data. Again, I'm very happy to take questions in the chat, or you're welcome to reach out to me any way that you'd like to and just discuss this. But hopefully that gave you a little more insight into what we've been up to talking about terroir and microbiomes.
Hi, my name's Chris Curtin. I'm an assistant professor at Oregon State University. Been working on Brettanomyces for a long time, and a persistent anecdotal idea has been that Brettanomyces exists in the vineyard. And I'm going to talk today about some research we've been doing to try and find out if that is indeed the case. So just by way of background, just to back up and make sure we're all on the same page when we're talking about Brettanomyces and its impact, what we know of as Brett taint. Or Brett taint or Brett spoilage is when we see these aromas and flavors such as Band-Aid, medicinal, and barnyard overpowering primary fruit flavors. And yeah, there's a place for these in terms of complexity, perhaps in some wines, but evidence suggests that consumers who don't know anything about these flavors respond negatively because ultimately they're detracting from what they're expecting, which is a nice fruit flavor in a red wine. So this is predominantly a red wine issue, of course. And the cause of Brett spoilage, or where this spoilage gets its name, is the yeast Brettanomyces bruxellensis. And what it does is it metabolizes natural grape precursors into volatile phenols, which are responsible for these aromas. The important thing to note is that these yeast and this spoilage occurs everywhere where anyone has looked at wine. So it's pretty safe to say that this yeast is ubiquitous. So this project really, or the core premise of this project, is to go and find Brettanomyces and try and work out where it's getting into wineries. So for a long time, people have studied Brettanomyces in the winery, and there have only been a few examples in the literature where researchers have identified Brettanomyces in the vineyard. And logically, if Brettanomyces is found all around the world in wine and in wine regions, a logical connection that all these places have are, of course, vineyards, as well as barrels and winemakers, and these things move around as well. But nevertheless, the focus of this project was about seeing if we could find Brettanomyces in the vineyard. Why would we want to do that, aside from knowing? It would obviously be helpful to know, maybe not at a single cluster level. If one cluster is affected by Brettanomyces and another isn't, it's unlikely to change processing decisions. But if we could, say, extrapolate this out to, well, this vineyard has a higher load of Brettanomyces on a typical year, or if certain viticultural practices are performed, it would be helpful to know that, so that you at least know going into processing that fruit whether there's higher risk, or if you can associate the prevalence of Brettanomyces in a vineyard with certain practices, then you can perhaps try and minimize the load that's coming into the winery. So if this is so easy to do, then why have so few people reported Brettanomyces in the vineyard in research? It's laborious to go through a process of going from grapes through to isolating yeast of any kind, let alone something as slow growing as Brettanomyces. But this is how we went about it, and this is based upon what other people have done, which ultimately is take grapes in the vineyard, do it aseptically so that we don't introduce any contamination. We do a step where we wash the grapes. Essentially what we're doing is washing the yeast and bacteria off the grapes into media that we can then try and find Brettanomyces in. And we resuspend what we wash off the grapes in media that encourages Brettanomyces growth. It's not super selective. It's very difficult to block the growth of all yeast. But the idea is to try and encourage the growth of Brettanomyces more so than others. So the media we used is something called BEM. Then we incubate that for a while, plate it out, and from that point on, the technology or the techniques are fairly straightforward in terms of identifying Brettanomyces. So the picture here down the bottom is just showing that process in general, and Kevin Pagao is the master student who did this work. And the samples we were taking were single cluster samples from 3 vineyards. So aside from this is a pilot study to try and see if we could isolate Brettanomyces from the vineyard, the other part we're interested in is how well this process works and what interferes with it, so that we can learn from that and try and design better protocols. So now I'm going to have a few slides, just 3 slides of data. And on the left-hand side is the data, and on the right are the key results. So if you don't want to look at the data, just read the results. But the main thing to take away from this slide is that we isolated yeast through enrichment. The results shown here are just from one vineyard because 2 of the vineyards that we sampled from in the first year that we did this, all the samples became heavily contaminated by mold and we couldn't recover any yeast. Brettanomyces is shown in red. So you can see that we did isolate some Brettanomyces, only in one of the years and not the other. And we isolated a bunch of other yeast species. The interesting thing is that 96% of these are what we'd consider rare vineyard species. We know that the vineyard is home to a lot of non-Saccharomyces yeast, but the yeast that we were isolating are not the ones that we typically would isolate if you just went out and plated a bunch of grapes. So what we're interested then is saying, well, okay, we know that we can isolate Brettanomyces, but there's a lot of other stuff coming through in these enrichments. Why are those yeast coming through, and how big of a problem is it? Does it explain why we're essentially only the 4th study in all of the grape and wine research that's gone on into wine microbiology? Our study is really only the 4th that's successfully reporting Brettanomyces isolation. This is one of the reasons why, because the non-Saccharomyces yeast, so on the left we have the data, the red line is showing the typical average for the Brettanomyces isolates that we were recovering and some that we were using as controls. So on the left-hand side of the plot is rate. That's how quickly the yeast or the cells are dividing, so how quickly the population increases. And several of the other yeast species were able to divide at a faster rate than Brettanomyces. On the right, we have something called growth delay, which is really a measurement of how long does it take for you to be able to tell that there's Brettanomyces growing. And in this case, what we see is that a lot of the other yeast have, in the darker blue color, have a shorter growth delay than Brettanomyces in this enrichment media. What this means is that they're going to show up earlier in the enrichment media. So if we're plating it out, then we're more likely to recover something else other than Brettanomyces. So we took this one step further. We then chose 2 representative species that we isolated from vineyards, Nakazawa isherwoodii and Kazakhstania aerobia, 2 not super common vineyard yeast. And we wanted to work out if we put them directly in the same media as Brettanomyces, who would win ultimately? And would that tell us about something about how successful we were likely to be continuing this kind of work? So in this case, if you look at the data on the left, an individual bar is representing a single tube where we've put into the mixture, into the enrichment media, an equal amount of 2 species. Brettanomyces alongside Kazakhstania, that's the aqua versus purple, and Brettanomyces versus Nakazawa, which is the yellow versus purple. So what we're looking for here is if enrichment's working, then purple would become dominant and we'd be more likely to isolate Brettanomyces. So if we fast forward to the most important part, which is when we played out, which is at 14 days, Brettanomyces was able to outcompete Kazakhstania, not so good at outcompeting Nakazawa. So that means if you had a bunch of grapes that had Brettanomyces and Nakazawa both present at equal populations, then we'd be more likely to isolate Nakazawa. On the other hand, if we had equal numbers of Brettanomyces and Kazakhstania on a bunch of grapes, we'd be more likely to isolate Brettanomyces. So in this way, we learned a little bit more about how the enrichment works and how it could be potentially made more successful, or at least work out which species, if they come through, that's possible they could be masking a Brettanomyces population on that sample. So in conclusion, it is possible to enrich for Brettanomyces, but this process mostly results in isolation of other rare vineyard yeast species, which they in and of themselves can be interesting. But if our focus is on Brettanomyces, it's just important to realize that it's very difficult to isolate Brettanomyces. Nevertheless, our pilot study does show that the vineyard could be an important point of origin. We just don't really know how widespread Brettanomyces is in the vineyard at this point, and how the populations that are in the vineyard might actually make their way, well, how they make their way, but how often do they make their way into the winery? So just to finish, I'll thank my lab, a very 2020 kind of photo there, and Kevin, who's the master's student who worked on this, James Osborne, my co-PI, and thank the Oregon Wine Board for funding this pilot study. And of course, all the wineries who were very helpful. Thank you.
Hello, my name's James Osborne. I'm an associate professor at Oregon State University and an enology extension specialist. And today I'm going to talk a little bit about a project we've been working on for the last couple of years, looking at malolactic fermentation and how it could be used as a tool to prevent or minimize the risk of Brettanomyces spoilage. So firstly, why did we undertake this research? Well, we know that Brettanomyces are a particularly problematic wine spoilage yeast that can cause spoilage to wine and is fairly ubiquitous in many different regions of the world. There's limited methods to control it. We know that SO2 can be used, but there's some differences in tolerance to SO2. I know pH and ethanol can also play a role, but it can be very difficult to control in the winery. And so we were looking at different methods or additional methods that might help. Brettanomyces spoilage can occur at a number of different places during wine production. But during or at the end of malolactic fermentation is a particular risky time for the wine where it is susceptible to Brettanomyces. This is because if you want to conduct a malolactic fermentation, you can't add SO2 to the wine. You have to wait until that process is finished to add SO2 to stabilize it from Brettanomyces. So the wine at this point is unprotected by SO2. Wine is usually kept in conditions that are conducive to encourage malolactic fermentation, so that's temperature-wise and maybe nutrients-wise, but that also makes it at risk for Brettanomyces spoilage. So you want malolactic fermentation to occur, so you keep your wine at maybe slightly above cellar temperature, but then that also makes it susceptible to Brett. That's one reason why a quick or vigorous malolactic fermentation is recommended, so that you can get SO2 on that wine quicker. So you don't necessarily want a long, drawn-out malolactic fermentation because that wine is at risk while that process is happening. Some additional new research in the last few years has also suggested another benefit of conducting malolactic fermentation. One report in literature, as well as some previous work in our lab, showed that the malolactic fermentation may actually suppress the growth of Brettanomyces. So that led to our main project goals for this. We wanted to further explore whether malolactic fermentation could provide additional protection against Brettanomyces. This is a process that's very common in winemaking. Are there ways maybe the winemaker could utilize malolactic fermentation either through different Enococcus strains or timing to maybe help minimize the risk of Brettanomyces spoilage? So we looked at strain differences, firstly Enococcus strains. So we used a number of different Enococcus strains, tested them against one Brettanomyces strain, and then we flipped the script and used one Enococcus strain and tested that against 8 different Brettanomyces strains. So are there strain differences both within Enococcus and within Brettanomyces bruxellensis? If there was inhibition, we were interested in what the inhibitory mechanism might be. If Enococcus was inhibiting Brett, how was it doing it? Because again, understanding that might help us if we're thinking about how we might manage the malolactic fermentation during winemaking. And then finally, we wanted to consider other wine stresses that we know can play a role in whether or not Brett causes spoilage. So ethanol and pH are 2 additional wine parameters that can cause additional stresses in the wine for microbes to grow. We wanted to look at malolactic fermentation as well as some of these other wine stresses. So what did we find out? Well, firstly, when we tested a lot of the Enococcus strains, we saw that they did inhibit the growth of Brettanomyces, one particular Brettanomyces strain, UCD 2049. So the way we conducted this was we put wine through malolactic fermentation. That same wine we put aside and had a portion that did not, so that was our control wine. And then at the end of malolactic fermentation, we inoculated that wine with Brettanomyces. So on screen here, we have a couple of graphs that demonstrate the inhibition of Brettanomyces. So here we have the control wine. So this line here, this is the growth of Brett if we inoculated into the wine that had not undergone malolactic. So we see pretty strong growth up above 10 to the 6. So you definitely have spoiled wine if Brett grew that well. Here, this line here is if the wine had first undergone malolactic fermentation and then we inoculated it with Brettanomyces. And you see quite a dramatic difference in Brettanomyces growth. In fact, we see a decline in population, and then for the rest of the study here, 50 days long incubation, we did not see any more growth of Brettanomyces. So there was clear inhibition of Brettanomyces growth. There was some minor differences between some of the strains. So some Enococcus strains, we saw a very strong inhibition like this. Other strains look more like this graph here on the right, where we still had that inhibition initially, but then we had the growth kind of slowly come back over time. So this told us that that inhibition occurred and that it was fairly, all of the strains that we tested inhibited Brett in this way. So our next question is, well, what's causing this inhibition? And so we designed an experiment to look at that. So this particular experiment was using a dialysis membrane to separate Brettanomyces cells from Enococcus cells. Now the reason we did this is if Enococcus was inhibiting Brettanomyces, it potentially could be doing it in 3 ways. One would be that Enococcus maybe consumes some essential nutrients in the wine, and the Brettanomyces, when you inoculate into that wine, doesn't have those nutrients, is unable to grow. A second scenario would be that Enococcus produced an inhibitory compound. So that inhibitory compound is in the wine, and again, when you inoculated Brettanomyces into that wine, it was inhibited. A third potential inhibitory mechanism is that just the physical contact of having a lot of Enococcus cells at the end of malolactic fermentation, adding Brettanomyces into that environment, there's the presence of a large amount of Enococcus cells inhibited the growth of Brettanomyces. That's called cell-to-cell contact. So this particular experimental setup allowed us to kind of figure out which of those mechanisms might be in play. So here on the far left, we have a situation where we have this vessel with Pinot Noir wine in it. We have within that another smaller vessel that has a dialysis membrane that allows the wine to flow and not restrict the movement of nutrients and not restrict the movement of any small compounds, any small inhibitory compounds Enococcus might produce. However, the pores in the dialysis membrane are small enough that Enococcus and Brettanomyces couldn't pass through. So this kind of physically separates the 2 organisms. So here's an example where we had both Brett and Enococcus in the same system. So they'd be physically in contact here. And then over on the far right here, we have where Enococcus is in the inner chamber and Brettanomyces is in the outer chamber. So this way, we're physically separating the 2 microorganisms, but any nutrients depleted by Enococcus would be the same in the wine on both the inner and outer chamber, or any inhibitory compounds produced by Enococcus would be the same on the inner or outer chamber. So what did we find out when we looked at our results? Well, just if we put Brettanomyces in the wine by itself, we got strong growth, as we'd seen previously. When we put both Enococcus and Brettanomyces together in that same wine, we again saw what we'd seen previously, that there was inhibition of Brettanomyces if we added it at the end of malolactic fermentation. But what about if we physically separated those cells? Well, that's where we saw this difference in that if we physically separated the cells, we all of a sudden got the strong growth of Brettanomyces back. So that told us that it wasn't production of a nutrient or it wasn't production of an inhibitory compound, because those compounds would pass easily through the system here, and we would expect to see the same kind of inhibition in this system here. So from this study, we concluded that that inhibition that we'd seen earlier was actually due to cell-cell contact. What about differences between Brett strains? Well, we'd seen previously with one Brett strain that we had inhibition, UCD 2049, here's that example again. But when we tested other Brettanomyces strains, we saw that the inhibition wasn't nearly as great as what we'd seen with the other strains. In fact, here's an example of the strain ODWI 1499, where we did not see inhibition by malolactic fermentation. So there did seem to be some strain differences. So the next piece was to figure out, next piece of the experiment is to try and figure out what that difference might be. One thought was perhaps differences in ethanol tolerance between the Brett strains might be causing these differences that we saw between Brett strains inhibition by malolactic fermentation. So we set up an experiment where we had lower or higher percentage of ethanol in the wines. We adjusted the wines and used those same Brett strains to see what happened. And in fact, that was what we found, was that Brett strains that were less tolerant to ethanol were more susceptible to inhibition by Enococcus, while Brett strains that were more ethanol tolerant, such as AWRI 1499, were less susceptible to inhibition by Enococcus. So there did seem to be this interplay between ethanol tolerance and malolactic fermentation. So what are our next steps? Well, we'd like to further explore this interaction between ethanol and malolactic fermentation inhibition. We'd also like to look at pH, because that's another parameter in wine that we know can impact Brett growth. And then finally, the timing of Brett infection during malolactic fermentation. All of the studies we've done to date had been, we conduct malolactic fermentation in the wine, and at the end of malolactic fermentation, we inoculate with Brett. But would we still see this inhibition if Brett infection occurred maybe at the beginning or midway through malolactic fermentation? So in practice, this might be if you had a barrel that was infected with Brett and you added fresh wine in there, inoculated from malolactic fermentation, would you get suppression of Brettanomyces in this case? So this work, I'd like to thank firstly the Oregon Wine Board for funding, and then a number of people who've helped work on this project, in particular Adam Lauderdale, the graduate student who conducted this work, Aubrey Dubois, the faculty research assistant in my lab, who also has helped with this work, as well as my collaborator Michael Chen, who's conducted volatile phenol analysis. Unfortunately, we haven't had time to talk about that today, but along with growth analysis, we also measured volatile phenol production by the Brett. Thank you.